All right, so it's 9 o'clock. So thanks everyone to logging, and then thanks everyone joining us for during this hour with us on today's museum seminar during the weekend, and particular big thank to our today's speaker, Ryan, my friend and my colleague, actually really sharing his recent work with us today. So it's absolutely my pleasure to welcome today's speaker, Dr. Ryan Yang from National Cancer Institute. As I said, he's been my neighbor and my colleague, my colleague and my friend for quite a while. So Ryan actually received his bachelor degree from Worcester Institute, where he completed his thesis work with Dr. Honghui Deng, and then afterwards, Ryan obtained his PhD in biochemistry from Cornell University, which followed with postdoc training at National Jewish Medical Research Center and National Cancer Institute. Later, after Ryan completed his postdoc training, he was later promoted to staff scientist in the lab of Dr. Lusto in NCI-CCR. So 2020, Ryan was appointed as a STEMN investigator in the lymph node malignancy branch in NCI, start his independent research program. So Ryan's lab is particularly interested in multiple myeloma, which is an incurable malignancy of plasma cells marked by extreme genetic and the phenotypic heterogeneity. So actually, I haven't heard about the multiple myeloma before I actually met Ryan. But afterwards, actually, I checked into a few literatures and I found it's actually quite fascinating. Multiple myeloma actually featured as the recurrent chromosome translocation involving in the immunoglobulin heavy chain locus primary which is primarily characterize this disease with a abundance of mutations targeting the RAS and NF-kappa B pathway, among others. So while the genetics of multiple myeloma has been well annotated, much less is known about pathogenic signaling in this disease or how common mutations may contribute to the oncogenetic signaling. So there's a lot of human immunology and then B-cell biology involved in Ryan's study, I think which is going to illustrate a little bit with us today. So Ryan's research program utilized cutting-edge proteogenomic techniques, high-resolution microscope imaging, and the biochemical approach to discover molecular machinery of pathogenic signaling in the multiple myeloma with the goal of finding new opportunities for the targeted treatment of this disease. Based on this, I think Ryan recently really actually complete a very, very fascinating study recently published on cancer cell, which I think he's going to share with us today. Really exciting to have you with us today, Ryan, and we are looking forward to your talk.
All right. Well, thank you for the very nice introduction, Sean. And hello, everyone. I'm excited to share our recent work on regulation of IRF4 in multiple myeloma. So, confession is, let's see here. I jumped ahead. I'm not an immunologist, and so I may be a little out of my element here. But I do study. I'm a cancer biologist, and I do study the cancers of the immune system, and specifically lymphoid malignancies. And so uh, it's kind of always been like a fascinating thing. If you, I think Tom Waldman, who was our former department chair, said if you're an immunologist, you study T cells. But if you're a cancer biologist, you study B cells, and there's something to that because it's sort of interesting because in in the human body, there's roughly like equal proportion of T cells and B cells out there. But if you look at malignancies, there's almost exclusively, at least the vast majority are within B cell lineages, such as lymphomas and what I'll talk about today, myelomas. And so it really comes all down to antibody production, which can be quite dangerous, as it turns out. And so depicted here is a is germinal center reaction, and so this is sort of where everything happens. And so a naive B cell, when it encounters its cognate antigen, will enter into this germinal center reaction. And I won't go through the details, except to say, as from a cancer biologist perspective, while in the germinal center, there's a couple major things that can happen to a cell to lead it down the pathway of malignancy. So first off is aberrant somatic hypermutation, which is what do I say, facilitated by aberrant AID activity. And this means you can have mutations outside of the immunoglobulin genes, but actually onto potential oncogenes or tumor suppressors. And the other thing that happens is that you can get chromosomal translocations in the immunoglobulin heavy chains. And this is because when you have class switch recombination, switch from IgM to another isotype, this gives an opportunity if something goes wrong to put a potential oncogene under the control of the powerful immunoglobulin enhancer genes. And so one of the outputs of, or one of the, if something goes wrong in the germinal center reaction, one possibility is multiple myeloma, which is the malignant plasma cell. And so multiple myeloma is, let me see, the second most common lymphoid malignancy following diffuse large B-cell lymphoma. I think overall it's about the 14th most common human cancer. In the US alone, it accounts for over 30,000 new diagnoses a year. And so on a cellular level, myeloma is descendant from or is akin to a normal plasma cell, and that it produces antibodies in the bone marrow, except there's just a lot more of these cells, and they continue to pump out prodigious amounts of antibody. And this leads to, unfortunately, all kinds of problems for patients, including renal insufficiency from all this antibody clogging up the kidneys. There are also pathological bone fractures caused by the expansion of these malignant plasma cells, and also these plasma cells crowd out the normal cells, so patients often develop anemia. And so, as Chuan alluded to, the genetics of multiple myeloma has been quite well studied. And one of the, actually, I think it may have been the first target of a next generation sequencing. Gosh, that was a long time ago now, probably like almost 13 years ago. And why is that? One thing is you don't need a surgeon to get samples. You can do a bone marrow biopsy. And so this is relatively easy. If not, it certainly isn't pain-free. But because of that, the genetics have been well annotated. And kind of another interesting feature of myeloma is because it's a plasma cell disease and they continue to secrete antibody, myeloma is often picked up incidentally from normal blood work because they'll notice a high amount of monoclonal antibody within the serum. And so as a result, you can really see this whole tumor progression happen. And so it starts off as something called monoclonal gammopathy of undetermined significance, a mouthful, also known as MGUS. And this is a pre-malignant state, but an expansion of plasma cells. This progresses to a malignant state called smoldering myeloma, which is basically over 10% of the bone marrow is plasma cells, but there's no obvious symptomatic disease. But once a patient develops symptoms, this is now full-blown multiple myeloma. And at the very end stage, you often get extramedullary disease, so outside the bone marrow, which is known as a plasma cell leukemia. And this would be the most aggressive and often end stage part of disease. And so there's a lot of genetic events. You can actually map these onto the developmental stage of the cell, which is sort of interesting. And a lot of the primary genetic events are generally ascribed to immunoglobulin translocations, either with regulators of the cell cycle or with regulators of transcription or perhaps signaling. There's also a lot of hyperdiploidy where you get trisomies of various chromosomes. And these seem to be somewhat mutually exclusive, which is interesting. Beyond these primary genetic events, you pick up many secondary events, and almost always you have MYC translocations and mutations of MYC that lead to enhanced MYC expression. You also get often deletions of P53, and also there's a lot of RAS pathway mutations. It's actually among the diseases, cancers out there. It's the third most, RAS mutations are the third most prevalent, right behind pancreatic cancer and colon cancer. And there's also almost all of these tumors find some way to activate NF-kappa B. So there's your myeloma primer. Uh, Good news is myeloma therapies have improved vastly over the decades. So initial therapies for myeloma were steroids, which do have quite an effect, but by themselves not so much. This is followed by alkylating agents such as melphalan, which was commonly used in the 70s. In the 80s, they went and were doing a lot of autologous stem cell transplants, which do prolong patient survival for patients who are eligible for these transplants, but most patients were not because they were certainly like many cancers, that's many of these patients are older and just can't tolerate this therapy. But there are some breakthroughs in the 2000s. So this is when people discovered the effectiveness of proteasome inhibitors. And so this would be like bortezomib or more recently carfilzomib. So these proteasome inhibitors basically target a plasma cell vulnerability, which is there's a lot of ER stress caused by the constant manufacturing and secretion of antibodies. And then also was the introduction of immunomodulatory drugs or IMiDs. These drugs include lenalidomide or pomalidomide, and these are commonly used as a mainstay of myeloma therapy. And these drugs lead to disruption of the oncogenic transcription networks underlying plasma cell biology and myeloma biology. More recently, there's been this massive explosion of immunotherapy in myeloma. So it started with monoclonal antibodies that target specific surface markers like CD38, so daratumumab. But more recently, there's been CAR T cells have been targeting surface antigens such as BCMA. And I think in most recently, you have these bispecific T cell engagers, which are also hitting the market. They've been FDA approved, and these are approved in patients who have failed all of these other therapies. However, with all of these therapies, myeloma does in fact remain incurable in almost every case, and all patients will eventually relapse and become refractory to further therapy. So, kind of the main one of the main questions we have in the lab is if we study, can we identify novel molecular vulnerabilities in this disease? And we were quite interested in a couple aspects of myeloma biology, pathogenic signaling, which I won't talk today about, but also oncogenic transcription networks. So the concept of the lab is we like to do large scale experiments. So we really do multi-omics experiments. And this encompasses CRISPR fitness screens to identify essential genes. We also do a lot of more targeted CRISPR screens where we're looking for a specific phenotype. And today I'll talk about knocking in GFP to gene loci and then using the expression of that specific gene as an endpoint for a CRISPR screen. We do a lot of spatial proteomics and also as well as global proteomics, and we also do global chromatin mapping. And the nice thing is there's a group, the MMRF, Multiple Myeloma Research Foundation, that has this publicly available data set of around 1,000 patients with all of their whole exome sequencing and RNA sequencing and clinical characteristics. So we can really filter all of our cell line derived data from these multi-omics screens to confirm that this is something relevant to a myeloma patient. And so filtering all of this, then we can discover these novel vulnerabilities. So everything in my lab starts with a CRISPR screen. And you can find a lot of myeloma cell lines in DepMap, and that is good data, but I'd like to say ours is a little better, mostly because we've taken a lot of care to express Cas9 that's very efficient within these myeloma cell lines. And we're also using a later single guide RNA library, the Brunello library, which has an enhanced activity compared to the Ivana library, which was used in a lot of these initial screens in the DEP map. Anyway, these screens are pretty simple. Basically, transduce Cas9 into these, select for exceptional activity, and once you have a stable cell line, we transduce with this Brunello guide RNA, whole genome targeting, 19,104 genes, I think. And it's pretty easy. You basically take a comparison of day 0 at the beginning assay, induce Cas9, and then compare the guide RNA frequency at day 21. So day 21 to day 0, we compute something called we call the CRISPR screen score, which is just a z-score of the dropout or enrichment of guide RNAs on a gene level. And so if we do all of this and then we filter out genes that are just essential in every type of cancer, we can see among all the genes in myeloma, IRF4 is the most essential gene in this disease on average. And this actually came as absolutely no surprise whatsoever, because it's already known that IRF4 is the master regulator of transcription, not just in malignant plasma cells, but in normal plasma cells. And this was really outlined by Lou Stout and Art Schaefer like 15 years ago or more in Nature. And it kind of they found this really nice relationship showing that IRF4 really cooperates with MYC, which is always aberrantly expressed in myeloma, it seems. And there's this basically this feedback loop where IRF4 and MYC kind of lead to increased expression of each other. And like I said, it leads to effectively this oncogenic transcriptional network that is imperative for myeloma survival. So we wanted to look at IRF4 because IRF4 is actually, it's a transcription factor. It's really, really hard to drug. Unfortunately, people have been trying. These immunomodulatory drugs, the IMiDs that I talked about, they do indirectly target IRF4, and they do this because they lead to degradation of Ikaros and Aiolos, which are two transcription factors that lead to increased IRF4 expression. And I would say resistance to IMiDs is probably like the primary clinical challenge in myeloma. And if you could directly target IRF4, you would really find a potential lifeline for these patients who become resistant to this drug. And so to explore this, we're interested in finding genes that may regulate IRF4 expression. And we wanted to be able to look at IRF4 expression in live cells. So to do that, we used a CRISPR knock-in strategy to put GFP just downstream of endogenous IRF4. So basically, this gave us the ability to see IRF4 as GFP signal. And we could do a CRISPR screen here, again, induce Cas9, 7 days of this to allow for gene deletion. And from that point, we FACS sorted and we just compared IRF4 GFP high cells versus GFP low cells compared to the total input. This allowed us to create a ranked list of genes that regulate IRF4 expression. And so what you'll see down here is if you have a negative score in this assay, knockout of that gene leads to a reduction in IRF4 expression. So the good news is if you knock out IRF4, guess what? You don't have IRF4 expression anymore. So the assay worked really well. And it's shown on this side over here. You can see we actually found well over 100 genes that seem to be involved in the substantially involved in regulation of IRF4 expression, which is to say expression of these genes is required in order to have IRF4 expression. However, there's all kinds of genes and there wasn't really much rhyme or reason, except a lot of them seem to just maybe generally control transcription or control translation. And so we didn't really know what to make of this list. And so in order to kind of narrow down potential hits, we went on and we performed an orthogonal proteomics approach where we identified IRF4 interaction partners. And our thought was, I don't know if it was a good thought, but it did work out, was something that that may directly regulate IRF4 expression may also directly associate with IRF4. So this experiment worked really well. And here's some sample data on this side. Actually, I should back up a second. For those of you who don't know, we use BioID2, And so BioID2 is a promiscuous biotin ligase. And so it's effectively about the same size as GFP. And just like GFP, you can just tag a protein with it. In this case, we tagged IRF4. And what it does is it can biotinylate acceptor sites within around a 10 nanometer radius. And so from there, you simply lyse your cells, perform a streptavidin pull down, and then enumerate your proteins by mass spectrometry. Um, Doing that, we could find actually, this is a pretty small interactome. We often see things where we get over 1,000 proteins, but in this case, it was 1 to 200 proteins. And again, an interesting list of genes, but what do we make of it? So the magic really happens when you take this multi-omic approach and basically just compare genes that are in these various data sets. And so what we did was we just made a Venn diagram, as you can see, and we looked at this CRISPR screen of that identified IRF4 regulators. We looked at this proteomics experiments that showed us IRF4 interaction partners, and we filtered that through genes that we identified as being essential for the growth and survival of myeloma. And when you do all that, you came down to 5 genes, and one of those being IRF4. So again, that's nice, it worked. The other one was EP300, which has actually been previously shown to control IRF4 expression. And these remaining 3 are actually all components of the SWE/SNF complex, which caught our eye, of course. So SWE/SNF is a chromatin remodeling complex, and basically it's an ATPA dependent reaction where it disrupts binding between histone and DNA. And this can lead to ejection of histones or sliding of histones. And all of this can then basically open up DNA to give it access to either transcription factors or other proteins that can either promote or inhibit transcription. And I should say that this chromatin remodeling complex comes in 3 main flavors. And so there's CBAF, and NCBAF and PBAF. And these all respond to different histone modifications. So it's noteworthy to know that ARID1A is a member of the CBAF complex, and the other 2 proteins, which were SMARCD1 and SMARCB1, are actually common members of all SWE/SNF complexes. So we were particularly interested in ARID1A and CBAF. So first, we have all this omics data, so now we actually have to validate it. And so just to show you, if you do knock down ARID1A, this is a knockdown experiment using shRNAs, but you can see a nice loss of IRF4 expression. So indeed, ARID1A is controlling IRF4 expression. And we also use a proximity ligation assay, or PLA, which is a method to look at co-localization of endogenously expressed proteins. And effectively, it's somewhat complicated, but it's a pretty cool technique, and you should look it up if you're interested. But the end result is you'll see a red dot wherever you see, in this case, IRF4 and ARID1A here are within about 40 nanometers of each other. And we also looked at other members of SWE/SNF in these cell line experiments, and we saw that these proteins interacted with each other in the nucleus. Moreover, we could see IRF4 and ARID1A had robust interactions, not just in myeloma cell lines, but in primary patient cases as well, as shown here. Again, suggesting that IRF4 and ARID1A do associate in the nucleus and on chromatin. And so we could look at this more directly, of course, using cut and run in order to globally map chromatin locations for all of these proteins here. So we looked at not just IRF4 and ARID1A, but also SMARCA4 and SMARCB1, which are shared members of SWE/SNF. And you can see in these 2, these are CD48 and SLAMF7 are 2 IRF4, known IRF4 dependent genes. We can see substantial overlap between IRF4 and SWE/SNF. But we can zoom out and look at a more global scale, as shown here with these tornado plots. And we map all the IRF4 peaks with the cut and run for ARID1A, SMARCB1, and SMARCA4, you can see that there's very similar patterns. And in fact, when you look at IRF4 peaks versus the ARID1A peaks, you can see that it's about 90% of those peaks are identical. There's a lot more ARID1A peaks than there are IRF4 peaks, but even under these ARID1A associated peaks, the IRF4 is the most enriched transcription factor. So this is all pretty interesting, but it turns out the role of ARID1A in cancer is pretty complicated, and it's actually generally viewed historically to be a tumor suppressor. And one reason is because there's a loss of function mutations in ARID1A that have been documented in several tumors, and perhaps it's very high frequency in ovarian and uterine cancers, as well as in colon and liver cancers. So we did analysis of ARID1A mutations in all the TCGA cohorts here compared to this MMRF COMPASS study. I think as you can see here in pink, there's almost no mutations targeting ARID1A in myeloma compared to the very high levels of mutations in again, these gynecological cancers or stomach cancers. And this really suggested that ARID1A is not acting as a tumor suppressor in myeloma. And in fact, if you look at DepMap, one major advantage of DepMap is that they have done over 1,000 cell lines from, I think, about 39 distinct tumor types. And so you can download all their data and look at their essentiality scores for when you break it down to these 39 different tumor types. And quite strikingly, you can see multiple myeloma is actually, among all of those, the most dependent on ARID1A, really saying that ARID1A is acting as an essential gene to drive myeloma pathogenesis. And so because this, usually I skip over this part, but because this is an immunology a seminar, this was a major component of the paper. And this was done by my friend and neighbor, Jagan Mupati. And what we looked at, we were wondering, does ARID1A have a role in normal plasma cells? And we really wondered that because I kind of alluded to this in the introduction, but many of the best therapies in myeloma actually target plasma cell vulnerabilities. And so, like I said, these proteasome inhibitors target the ER stress from all this antibody production, and these IMiDs are targeting this oncogenic transcription. So we wondered, is ARID1A just part of normal plasma cell biology? And so to look at this, what Juggin did was we actually got from Mike Leonardo these ARID1A mice. They were ready to cull them to eliminate the colony that week. So I was lucky in that regard, and I was able to get these mice. So Juggin took them and he bred them with a AID-CRE so we could make a conditional knockout in germinal center B cells. So to produce germinal centers, he did a sheep red blood cell induction of germinal centers twice, and then waited 8 days, at which point, by fax, plasma cells were enumerated from spleen, Peyer's patches, and bone marrow. So we saw that ARID1A is in fact necessary for plasma cell formation, and we see a significant decrease in plasma cell numbers in spleen, Peyer's patch, and bone marrow, actually most strikingly in bone marrow. But the one thing studying plasma cells in these different anatomical sites is you can see there's basically hardly any plasma cells. They're very low frequency in these various organs. And so to kind of convince ourselves that these numbers really were real, we also looked in the lamina propria because you have much higher concentrations of, in this case, actually IgA plasma cells. And so what you see here is, again, a very significant reduction in the number of plasma cells upon ARID1A knockout in germinal center B cells. And you can see this not just by fax, but you can nicely image these plasma cells in either littermate control or ARID1A knockout mice using IgA, which would be in the plasma cells, as shown in green here. And you can see a nice reduction in both the ileum and the duodenum. Of course, one possibility is that there's just less plasma cells in ARID1A knockout mice because there's less germinal center B cells. So of course we looked at that as well. And truth be told, we did see a reduction, or say a trend towards reduction in germinal center B cells in both the spleen and Peyer's patches, but it didn't raise to the level of significance, suggesting that ARID1A had some role perhaps in germinal center B cells, but perhaps a more potent role in the plasma cell. And to kind of look at that a little further, we did ARID1A cut and run in mouse B cell subsets that we purified by using cell sorting from these mice, from wild type mice. And so we looked at naive germinal center B cells and plasma cell B cells, and we looked at ARID1 cut and run. So we could see ARID1A chromosomal location of the protein. And if we look at these three plasma cell specific genes, so XBPP1, PRDM1, and SDC1, you can see in pink here that you see lots of ARID1As decorating these genes. But interestingly, we also saw a lot of ARID1A binding for germinal center specific genes, and actually even such as AID here, and also even for naive B cells. And so again, we can look at this in more of a global level, and this is a little complicated, but basically what you can see is plasma cell specific peaks, and we can map them onto what we saw in naive cells, germinal center B cells, or plasma cells. And you can see like a stepwise increase in ARID1A binding kind of globally. However, you can also see that there's plenty of binding in germinal center B cells as well as in naive and cells as well, suggesting that ARID1A has more of a, I guess, global yet somewhat nuanced role because it can cooperate with many potential lineage-specific transcription factors. But interesting, in plasma cells, we do actually see that IRF4 is one of the top enriched transcription factor motifs for ARID1A. So again, confirming that ARID1A and IRF4 have a special connection. So somewhat fortuitously, we had begun these studies, and around just a few months later, actually, a couple SWE/SNF inhibitors became available. So the first one is an AU15 is a SMARCA2/4 degrader. So SMARCA2/4 are components of the A2PA subunit, so they're necessary for activity of all SWE/SNF complexes, so not just CBAF, but also NCBAF and PBAF. And so this is a degrader, AU15330. And then this company, Foghorn Therapeutics in Boston, later released an actual chemical inhibitor. And this is now, it's definitely been through a couple of phase 1. I think it's actually in phase 2 trials now. And so they're available in the clinic. So we first had typo. We first had used this AU15330 and to examine basically how does this targeting SWE/SNF affect chromatin accessibility in myeloma. So to do this, we did attack-seq, and we did 2 cell lines, either this SKMM1 or H929, 2 classic myeloma cell lines, as well as a primary myeloma patient. And what you can see here is looking at DMSO versus drug treated, we can see that there's this very rapid, complete closing of, not complete, but large closing of the myeloma chromatin. And you can see this in every case. And interestingly, you can look at these genes that are significantly downregulated, and you can map them onto the IRF4 cut and run data. It's the same peaks, I guess, not genes, but on a peak level. And what you can see is that really for the IRF4 dependent genes, you can see that there's this huge reduction in chromatin accessibility, saying that SWE/SNF really seems to preferentially target the IRF4 dependent transcription. And you can see this perhaps even more directly using RNA-seq to look at changes in gene expression upon treatment with this degrader AU15330. And so what we see is when you inhibit sweet-SNF, you get this massive and very significant, ridiculously significant reduction in IRF4 gene expression. And kind of one other thing to note is that you also see a very good decrease in MYC expression. And I'll get back to that in just a little bit, but something to keep in mind. We also can see CD48 and SLAMF7, which I noted were IRF4 dependent genes. And so besides seeing this by RNA-seq, we can also look at reduction of IRF4 protein by FACS analysis in primary myeloma cells. And what you see again is a dose-dependent decrease in IRF4 expression upon treatment with either this sweet-SNF degrader or sweet-SNF inhibitor. And the faces here are blocking the data, but I think this is lenalidomide over here, where you see no reduction. Oops. Likewise, we can look by Western blot with either the degrader or with the inhibitor, and what we see is this rapid reduction of IRF4 expression. And you also see quite a nice reduction in MYC expression. And so keep in mind that IRF4 and MYC do actually control expression of each other, but it turns out it may be a little more complicated than that. And so we did the IRF4 cut and run previously, and we'd also done sweet-SNF cut and run for various things, ARID1A, SMARCA4, etc. We now did a MYC cut and run, so we could map MYC locations throughout the chromatin, and we kind of saw two discrete subgroups here. So MYC kind of overlaps with pretty much everything, which is to be expected given its role as a general, let's say, amplifier of transcription. But we can see that there's peaks where you have IRF4, MYC, and sweet-SNF, or you just have sweet-SNF and MYC. And we kind of were curious, so we looked at what's going on there. So when you look at the IRF4 picks, and this is this LOLA analysis, and I won't go into too much detail on this except to say that you can see under the IRF4, you get this, looking at darker things here, you get transcription factors related to things you expect with IRF4, such as Aeolus and NF-kappa B, show up under these peaks here. Conversely, under these peaks, which are just MYC and sweet-SNF, you see things that are generally related to amplification or just general transcriptional regulation. And again, you can actually see a differential distribution of these peaks, whereas the MYC peaks really just settle right over the transcription start site as would be expected, whereas the IRF4 peaks are kind of decorated. So it almost seems like you have two independent things happening down when you inhibit sweet-SNF. And this actually matched data that we mined out of the Broad Institute's DepMap data, where first off, we took all the genes that were important for CBAF, PBAF, or NCBAF. We made an average of the Cronus score, so this gene essentiality score, and we made this ranked list here. And again, you can see myeloma is actually the most sweet-SNF dependent malignancy, actually next to diffuse large B-cell lymphoma. But quite interestingly, you can see that it's not just CBAF they rely on, but also heavily on PBAF. And there had been studies previously showing that MYC activity is really dependent on PBAF activity in myeloma. And I think what's going on is that you have, by inhibiting all sweet-SNF, you're actually getting kind of a 1-2 punch where you get both IRF4 and downregulation and MYC downregulation through separate BAF complexes. And in the case for myeloma, this is perfect because they're highly dependent on both IRF4 and MYC. So of course, the obvious question next is sweet-SNF inhibition toxic to myeloma? And of course it is, as you are not surprised to hear, since it reduces or it interrupts both IRF4 and MYC. And you can see we looked at a few different types of cells, but in the pink here is myeloma, and they were all generally sensitive to inhibition with this FHD286, which is the SMARCA2/4 inhibitor. And they were on average more sensitive than, this is germinal center B-cell lymphoma cells, and then this is activated B-cell DLBCL cells. And you can see we looked at, as a control, just a small panel of adenocarcinomas, which had very little activity. So among all these cell types, myeloma was the most sensitive. Kind of a notable thing here in red is that these cells actually have ARID1A mutations, loss of function ARID1A mutations. And in both myeloma, there's one cell line that has it. You can see that it doesn't actually affect its sensitivity to the drug. GCBs were also really sensitive to this drug, and this was actually recently published by Ari Melnick's group, also in Cancer Cell. And I presume this is really has to be chalked up to the fact that it's known that GCB is also highly dependent on MYC for their pathogenesis, but we haven't looked into that further. So it's not just cell lines, which is over here, but we also, again, we could look in primary myeloma cells, and you can see a really nice concentration, a dose-dependent induction of apoptosis as measured by annexin 5 in these cells, really suggesting that this might be clinically relevant. And one concern we had is that if you're inhibiting SWE/SNF in global regulation of chromatin accessibility, what's going to happen to an animal or a human if you do this? I said FHD-286 has actually been in clinical trials, so you can find a dose. And we want to see it for our own eyes, of course. So we did a xenograft in this case, and 2 doses of this FHD, which are both pretty low because it is known that if you go too high, you will get some toxicity. But we could generally control tumor growth in the xenograft setting without too much toxicity. Although we'll say at the higher dose, even though it did suppress tumor, we discontinued treatment at 2 weeks because the mice were starting to lose weight. So we also use this drug to generate a gene signature of SWE/SNF activity. And so basically this signature incorporates 93 genes. And we could take this signature and apply it to this MMRF Compass data set, which has around, I think there's about 750 patients that have RNA-seq data. So basically, we took the average expression of those 93 genes among these patients and ranked them by quartile. And you can see if you have high expression of these genes that are SWE/SNF dependent, you have much worse prognosis than than patients with lower expression of those genes. And it was highly significant, as shown here by Cox proportional hazard analysis. And it wasn't just the SWE/SNF signature didn't just track with patient outcome, but basically any metric of aggressive myeloma that you could think about, we could see a nice correlation between that. And so I won't get into too many details, but myeloma, it can be broken up into various subsets based on its gene expression. But this PR stands for proliferation subset. And among all the subsets, that's the most aggressive one. And I think you can see it's like heads and shoulders has more gene expression for the SWE/SNF signature than all the other samples. Moreover, P53 expression or deletion is highly correlated with aggressive disease. And you can see that this signature is correlated to that as well. Finally, over here, I've shown disease state. If you go from MGUS to myeloma to plasma cell leukemia, you can see a stepwise increase of this signature. Notably, this PR subset, is based off of 50 genes, and it is known to be correlated with aggressive myeloma, but there's effectively no overlap between this PR subset and the SWE/SNF gene signature that we created, suggesting we found a completely independent way to identify aggressive myeloma. So want to know, basically, I think I'd said earlier that lenalidomide, which is an IBID and can target IRF4 indirectly, resistance to that drug is a huge problem, and that's one of the major hallmarks of resistance to therapy. And so we want to know, could this drug treat cells or potentially patients who are resistant to lenalidomide? And so I'll get into that, but we generated cells that are resistant to lenalidomide just by increasing doses over months for these cells. And so we have MM1S and H929, which are two of the myeloma cell lines, which are naturally sensitive to lenalidomide. And so you can see it worked quite well. Over here is a viability assay with lenalidomide, and you can see that we were able to generate cells that were no longer sensitive. However, I think you can see here, if we treat with this FHD286 SNF inhibitor, it doesn't matter. All of these cells die, really suggesting that are still able to suppress IRF4 expression. And of course, by Western blot analysis, I can guarantee that we are. Up here is lenalidomide sensitive cells. If we treat with either the degrader or this FHD inhibitor, you can see really nice suppression of IRF4 in either lenalidomide sensitive or resistant cells. You just crush IRF4 expression. Interestingly, though, with lenalidomide shown here or here, you do see in the sensitive cells a nice, but much smaller decrease in IR4 expression, which completely just disappears when you make lenalidomide resistant cells, showing that SNF inhibitors can really still target IR4 expression. And not only that, but in fact, in these cells, these three cell lines are actually innately resistant to lenalidomide. But if you treat with lenalidomide and FHD, you actually see a very nice synergy, suggesting that you can actually get pretty good activity even in cells that are just naturally resistant. And which got us thinking, we thought you can add lenalidomide to this SNF inhibitor, but what else can you do as well? And so we're fortunate to have a great long-term collaborators, Doctors Craig Thomas and Michele Ceribelli, who are at the NIH NCATS, and they have a facility where they can do high throughput combinatorial drug assays. And so they have a library of over 2,800 agents, which are either in clinical trials or actually approved by FDA or about to be entered into clinical trials. And they're mechanistically annotated. And what you can do is you can look at your favorite drug. In this case, it's going to be FHD286 versus these 2,800 compounds, and you can look for drug synergy or antagonism. And so we did this with them. And we just did this in two cell lines because it takes quite a bit of work on their behalf. But we can see on this side, we saw many drugs that came up as being highly synergistic in both of these cell lines with FHD286. And really interestingly was that the RAS pathway was way overrepresented in these hits. So you could see both MAP kinase, classical MAP kinase inhibitors like MEK inhibitors and ERK inhibitors. You could also see mTORC1 inhibitors, which we know is downstream of oncogenic RAS and myeloma. And as well as actually one of these cell lines has a therapeutically actionable KRAS G12C inhibitor, or G12C mutation with an inhibitor that can target it. And that also showed up really strongly. And so RAS is sort of an interesting pathway for myeloma because in newly diagnosed myeloma, about 40% of them have KRAS and NRAS mutations. And RAS is my other favorite subject, but I won't get too much into it. But in a relapse refractory setting, you actually get upwards of 70% of tumors have RAS or NRAS mutations. So it's quite relevant in this relapse refractory setting that we're most interested in. And we could confirm that there was great synergy in RAS-dependent cell lines, but not RAS-independent cell lines. And so the mechanism behind this isn't quite clear, but we could see this very rapid induction of apoptosis when you combined FHD286 with a MEK inhibitor, trametinib in this case, whereas at 40 hours, we usually have to wait at least another day to see good apoptosis with FHD alone. And actually, these cells are innately just resistant to MEK inhibitors, which is, again, interesting, but a completely different story. So this combination induced rapid and really massive apoptosis. And again, when we treated mice with this combination, we could see almost a complete quenching of tumor growth in these mice compared to single agents. And it should be noted that at this concentration, these drugs were actually very well tolerated. So that's it. I just want to summarize that what I've shown you today is IRF4 is the prime vulnerability and target for drugs in myeloma. It's just not been possible to drug it. So we looked at this and we found IRF4 is really interconnected with ARID1A, which is this expression is dependent on that, interacts with it physically, and that they are on chromatin. And so treatment with sweet-SNF inhibitors just curtails IR4 transcription networks, which induces potent toxicity to myeloma, both in vitro and vivo. And we found that combination therapies of the sweet-SNF inhibitors with RAF pathway inhibitors is highly synergistic. And with that, I'd like to just thank everybody in the lab. And I want to do a particular call out to Arnold Bolomsky, who really spearheaded, just completely drove this project. And also our mass spec facility from Thorkel Anderson and Ronald Holowinski, as well as Jagan Mupati and people in his lab who did all of the work with the ARID1A knockout mice, as well as Tony for doing xenografts, the bioinformatics team at NCI, and the clinical team for providing samples. And with that, I'm happy to take any questions.